Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
So in this chapter, we're going to be talking about microbial growth. And in that little video you just saw, it illustrates that when I talk about microbial growth, I'm not talking about the growth of an individual cell, but I'm talking about the growth of a bacterial population. So just like you saw in that video, when bacteria are growing as a population, like what we have on a petri dish here, one bacterial cell, we can put one bacterial cell on the petri dish, and then over time, that one cell is going to divide and divide and divide, and then you're going to have a colony if you're on solid media, and that colony could have hundreds of millions of bacteria. So bacteria grow very quickly, and in this unit we're going to talk about what bacteria need to grow and what are some of the limiting things that influence them. So there you go. That's where we're going with this. First learning outcome I have is state the importance of the elements CHONPS, chomps, to bacterial cells and how they are acquired by cells. And so here's the periodic table of elements. And if you look at this pie chart, it's showing you that if you take bacteria and you analyze it, it's going to be 47%, or almost half of it's going to be carbon. Another third is going to be oxygen, there's hydrogen, and there's your nitrogen, phosphorus, and sulfur, and then some other things. So, but the primary elements are shown here. And this is true for all living organisms. You know, we're a carbon-based life form. So carbon is really important. And so I'm not going to talk about all these nutrients and how they're acquired by cells, but in particular in this series of lectures, we're going to talk a little bit about carbon, a little bit about oxygen, and go from there. Distinguish between the autotrophs and the heterotrophs. So when we're talking about autotrophs and heterotrophs, we're talking about how do bacteria acquire their carbon. And not just bacteria, I'm talking about bacteria today, but all living organisms can be classified as autotrophs or heterotrophs. So when we're talking about heterotrophs, we think about hetero means different, troph refers to eating. So they eat something different. They use organic molecules as a carbon source. Use organic molecules as a carbon source. These are the organisms that when we look in ecology and we're looking at food webs, these are the organisms that consume things, consume other organisms. And so you can imagine that humans are heterotrophs. There's plenty of bacteria that are heterotrophs, and lots of animals that are heterotrophs. A lot of living organisms in all different categories are heterotrophs. So they use organic molecules as a carbon source. So when we need carbon, we eat food. We eat carbohydrates, we eat proteins, we eat lipids, and they provide us not only with energy, but they're our source of carbon. So we get it from something other than ourselves, hetero. When we're talking about autotrophs, these are the organisms that use carbon dioxide as their carbon source. And these are the producers. These are the photosynthetic organisms. And probably when you think of autotrophs, if you have any experience with that, you probably think of plants. So plants use energy from the sun and carbon dioxide in particular, from the air. And they make their own carbohydrates. So they are autotrophs, or they're self-feeders. They make their own organic molecules from the carbon dioxide in the air. And so autotrophs, like I said, are the photosynthetic organisms. They're plants, but there's a lot of prokaryotic autotrophs also. One of the big categories of prokaryotic autotrophs are the cyanobacteria. And a lot of times you hear about cyanobacteria when you're hearing about algae overgrowth in ponds, and even in the ocean it can happen. Ponds, lakes, other waterways. So cyanobacteria are the autotrophic prokaryotes. And here's a river or a lake or something that has an overgrowth of of cyanobacteria. Usually this happens when there's too much nutrient in the water. There's pollution in that water, sometimes farm runoff or other sources of nitrogen and phosphorus that encourage these organisms to grow. But the photosynthetic prokaryotes, a lot of those are in groups called cyanobacteria. Here's a microscopic picture of them. You might have heard these called blue-green algae, same thing. There actually are microscopic algae that are also eukaryotes. So sometimes you hear about toxic algae that's made of due to a eukaryotic growth of microbes, an overgrowth of eukaryotic microbes. But these would all be autotrophs. So these are photosynthetic organisms. Most of the bacteria that we associate with every day are heterotrophs. They're, the things growing on our bodies. They don't need sunlight to grow. They use the food that they acquire in and on our bodies. So the next thing your book talks about is diffusion and osmosis. I think it's kind of in a little bit of an odd place, but diffusion and osmosis are important when you're talking about microbial growth and control of microbial growth. And so I want to just talk about it a little bit. A lot of you have probably heard of this, and I hope to get everybody on the same page of having a good understanding. A lot of the molecules that cells use, whether you're bacterial cells or any other kind of cells, diffuse into the cells. They move from high concentration outside the cell into lower concentration into the cell. So we've heard of this word diffusion, and it simply means the movement of molecules from high concentration to low until they reach an equilibrium. And so here's a classic example of diffusion. We have a beaker of water, you drop a drop of dye in it, wait a little bit, and you can see it kind of spreading out. Molecules and atoms, they have kind of a natural vibration going on. And they bump into each other and they cause this dye to spread out. And that dye is going to spread out until it's uniform, until we've reached an equilibrium. And so this is the idea of diffusion. And this is happening with cells all the time. Molecules can diffuse in and out. It happens in a room when somebody's wearing perfume, and it can diffuse from that person throughout the room. It happens in all different kinds of ways. Osmosis is a special kind of diffusion. And here it says osmosis is the diffusion of water. Oh, so wait, let's remind ourselves of that definition. The movement of water from high concentration to low. So the diffusion of water, or the movement of water from high concentration to low. That's one thing to notice about osmosis. We're only talking about the movement of water through a semi-permeable membrane. So wait, membrane was that, remember we had that phospholipid bilayer on the outside of our cells. So that's a membrane. And what we mean to be semi-permeable is that some molecules can go through and some don't. And plasma membranes, also known as cell membranes, are semi-permeable membranes. If they weren't, the inside of the cell and the outside of the cell would look the same and we wouldn't need cells. So osmosis, the diffusion of water, meaning the movement of water from high concentration to low until you get to equilibrium through a semi-permeable membrane. So what we're talking about is how water moves into and out of cells. And this has a big impact on where bacteria can grow or where any cells can grow, and what conditions favor their growth. So before I go on to a little bit more about this, I just wanted to remind you of some vocabulary that you might be familiar with. We have the word solvent, that in biological systems is water. Water's the thing that does the dissolving, the solvent. The solute is what is dissolved. So we have the solvent water, and the solute, in this case, in cells, we're going to talk about usually about salt or sugar when we're talking about osmosis. Could be proteins, but anyway, anything that's dissolved in there. And so when we have a solution, it's the solvent with the dissolved solute. So if I had salt water, the solvent would be water, the solute would be salt, and the solution would be salt water. So what we're going to look at over here are the natural conditions cells are faced with living in. And so in this example, this little thing right here is just a cartoon cell. And they're telling you that inside of this cell, there's 20% solute. So there's 20% sugar or salt or whatever it is. 20% solute. That, if we bring this to 100%, that means we have 80% water in there. Now in this picture, the little green dots are the solute, like the sugar. Now this cell was placed in a solution containing 20% sugar, solute, and 80% water. It was the inside of the cell and the outside of the cell had the same concentration. They've got the same solution. So this would be called an isotonic solution. Iso means the same. So we have, and so according to the idea of osmosis, it tells us water, we always talk about water, movement of water. Water is going to move from high concentration to low. Oh, it's a semi-permeable membrane. There's the plasma membrane. And in our example here, the solute, the salt or sugar, can't get across the membrane. It's stuck in it where it is. So diffusion of water, let's see. So we're saying 80% water on the outside, 80% water on the inside. So for every molecule of water that moves out, one molecule is going to move in. And the cell's going to maintain its natural shape, and it's going to do just fine. That's a really good situation for a cell to be in. Inside of our bodies, in the human body, our urinary system is always balancing the amount of salt and the amount of water in our bodies, and we retain water, or we'll urinate more, or we will retain salt or lose more in our urine depending on what we've eaten. And our kidneys are always testing the concentration of the solutions around our cells, so the concentration of our fluids in our body. And so we need to maintain an isotonic solution in our bodies for our cells to work normally. If we take that same cell, so here's the cell, the green dots are the solute. We have 20% solute, 80% water inside the cell, but we have a higher amount of solute on the outside in this example. So there's lots more of these green dots. In this example, they took 40% solute on the outside, which gave us 60% water to get us to 100%. Osmosis says water is going to move from high concentration to low. So 80% water on the inside, 60% on the outside. In this situation, water is going to move out of the cell, trying to achieve an equilibrium. The water is going to move until you get the same concentration of the water on both sides of the cell. Now, what's going to happen to this cell, because it's losing water, the cell's going to shrivel up. When we put a cell in a solution that has a high amount of solute and therefore a low amount of water, we call that a hypertonic solution. A hypertonic solution is a solution that has a relatively high amount of solute, making it have a lower amount of water. And that's going to cause a cell to shrivel up. This is bad for cells. Cells need water inside of them, and they need a good amount of water to carry out all those chemical reactions of metabolism. So putting cells in hypertonic solutions is not a good thing if you want to keep these cells alive. That's deadly to cells. The last thing we can do to that cell, and we have the same cell here, we have that cell with 20% solute, 80% water. In this situation, they took that cell and they put it in a solution with 10% solute, and that will make it up to 90% water. Osmosis says the diffusion of water through a semi-permeable membrane. Water is going to move from high concentration outside the cell to lower concentration inside the cell. What happens is that cell will keep acquiring water. Water will get absorbed into that cell. And so what's going to happen is that cell is going to get larger. And you can see this cell looks a little bit larger than the one here. Now, if this is a human cell, that cell could very likely absorb enough water that the cell will actually pop. And that can kill cells. So when we put a cell in a solution that has a relatively low amount of solute, that makes it a hypotonic solution. Hypo means low amount of solute, and that's going to cause the cell to expand and eventually lyse, which means burst. Now, this is what it looks like when you're talking about animal cells. But you probably are quite aware that you have your houseplants and you water them. Oh, maybe it's wilted because it's running out of water. Oh, the cells are kind of shriveling up. We know that can kill it, right? So you water it, and all of a sudden it's got a lot of water, and the cells start swelling up. But your plant never explodes. You can give it water, but it never explodes. Well, the plant cells have cell walls. Remember, all cells have a plasma membrane, that phospholipid bilayer. But plants and animals and some other organisms have, plants and bacteria, as well as some microscopic eukaryotes, have an additional layer that's a cell wall. Animal cells do not have a cell wall. Our own cells will burst in a hypotonic solution. Plant cells just get nice and firm. So think about how your plant, the difference between your wilted plant, the cells are shriveling up, you water it, and the cells get nice and firm. And that's because this plasma membrane presses against the cell wall, and now you've got nice firm cells. The same thing happens to bacteria. Bacteria have that nice cell wall that has that peptidoglycan in it. So you put bacterial cells in a hypotonic environment, they're fine. They're doing just fine. So plants and bacteria can survive hypotonic solutions. Animal cells can't. Oh, I thought I had something else on that. Oh, I just want to remind it. Okay. The other thing I want to remind you about osmosis, if that all seems kind of like I don't get it at all, I did another small video just on osmosis and put it on Brightspace. So if you want to watch that, in addition to this, you can. You don't need to, but if this doesn't make sense, I would do that if I were you. I'm going to stop right here and call it the end of this video.